Start recording. Good morning, dear student. This is your Professor Carver. Remember that 14 this chapters, Chemistry of Life, is also common with 1408, which is non-major biology. So we are going to finish the Chemistry of Life, our chapters, and we talk about all the macromolecules. We talk about, if you remember very well, we talk about the structure. First, we talk about the chemical groups, the carbonyl group, carboxyl group, amino group, phosphate group, sulfide group, methyl group. And then in the part 2, we talk about the structure and function of macromolecules, carbohydrate, lipids, protein, and nucleic acid. We didn't talk about protein and nucleic acid yet. We talk about carbohydrate and how carbohydrate is a polymer that we call the polysaccharide. What is a polymer? Is a big molecule that is made. It's a macro, big. Macro means huge. Macromolecule that is made of micromolecules that they are repetitive units. Like for example, starch is a repetitive unit of glucose, and they are bounded by glycosidic linkage. Glucose also can make disaccharide when they are only two sugar bonding together. The monosaccharide are those repetitive units, are those monomer, are what we call the micromolecules. Then we talk about lipids. Lipids and the polysaccharide, you have to know their function. They can be storage functions or structural functions. Storage, such as a starch for plant and glycogen for animals. Structures, the one that make a structure, which is a cell wall in the plant, which is the cellulose. Then we talk about lipids, and lipids are hydrophobic, and their function is to store energy, right? And the lipids are not a polymer. They are not, because they are no repetitive unit. They are not made of micromolecule that repeat itself, right? They can be a triglyceraldehyde, which is a molecule of glycerol that have a 3-hydroxyl group that's bonded to fatty acid. And I explained to you that anytime we have a fatty acid, the word acid, that means the functional group on it, it's a carboxyl. If I said amino acid, that means there are two functional groups, one amino group and as carboxyl group. If I said hydroxyl, that means it have an OH. And all the time when they are an OH, it's all like glycerol. This glycerol, it's a 3 hydroxyl group. And we can make 3 glyceraldehyde, 3 glycerides, or monoglyceride or diglyceride. Depend how many fatty acids are going to be bonding to this hydroxyl group. The hydroxyl group will give me the hydrogen, and the carboxyl group will give me the OH to make a big molecule of a lipid, which is a monoglyceride. If it's bound only to one fatty acid, if it's bound to 3, like here these 3 fatty acids, they are identical, they are the same. So all of that H, this hydroxyl group will give me H, and they will bond to the OH. This H will bond to this OH, and this H will bond to this. I don't see this. So this is this H for the hydroxyl group will bond with the entire hydroxyl group of the carboxyl. And this H, the second fatty acid. And this one, the last fatty acid. This will make a big molecule of a fat that we called triglycerides, because there are 3 fatty acids with the hydroxyl group. And those fatty acids can be saturated or unsaturated, all of them, or mixed. What is saturated? When at least one double bonding exists between carbon and carbon, this is unsaturated. One, even if it's big, huge molecule, and one double bonding exists, that's it. The molecule, it's called unsaturated fatty acid. This one doesn't have any carbon-carbon double bonding, so it is saturated fatty acid. And then we talk about how unsaturated, saturated fat, they have, all of them have single bond carbon-carbon, and they are very close to each other. Therefore, they are very close to each other because it doesn't have another step, single bond. So they are very close to each other. So at room temperature, these make them solid, like the fat here in this piece of meat. And those, the other one that they have a double bonding, it makes them really not packed closer to each other because they are like a second step. It's like me and you, between us, it's only one, it's only one step. And the other one, they are like more. So the one that they are like 2 steps between me and you, that's much, we are not closer. Double bonding, it's like you're putting another step in the top of the first step. So it makes it not packed closely. And therefore, more double bonding, more this molecule is liquid at room temperature. And this is the think of olive oil. All right. Another fat is the phospholipid, which is the major constituent for the plasma membrane. This fat have one glycerol in the head and the phosphate group. If you see, they have a glycerol, right? And they have the phosphate group. This head that is here in orange, it's a hydrophilic, it interacts with water. But the 2 tails, they are hydrophobic tails. They can be either saturated or unsaturated, one saturated, the other one unsaturated, both of them unsaturated, or both of them saturated, depend which kind of fatty acid it is. And this is the major constituent for the plasma membrane. And the plasma membrane, it's a phospholipid bilayer. That means it's made of 2 layers, 1 layer over here and 1 layer over here. And this property that is made of this hydrophobic part, hydrophobic, make the plasma membrane very, very selective. It controls what is coming in and what is going out of the cell. You have also other lipids. You see, this is no repetitive unit. This is by itself here. By itself, this is a molecule of lipid. This is another molecule of lipids, all right? They make the plasma membrane, but they are not making like a molecule that we call, I don't know. Lipid is not a polymer, okay? This is, for example, it's a lipid. By itself, it's a lipid. This is what we call phospholipid, all right? This over here, it's a phospholipid. It's true. This is a glycerol with a fatty acid, and the bonding over here, it's called ester bonding between glycerol and fatty acid. It's called ester bonding. Anyway, they have also some ring that they are fused. When they are like 4 rings fused together, this is steroids. And some of the steroids can be either sex hormones, or cholesterol, or cortisol. You see, they are all made of a ring, fused ring together, 4. This is very typical for lipids. All right. So now let's talk about another macromolecule that is a polymer. Proteins. Proteins can be found in cheese, meat, fish, eggs, milk, yogurt, right? It's a polymer. Why? Because it's made of repetitive units that we call amino acid. There are like 20 amino acid. And the function is of the lipid, they can play a role as transporter. Look at the transporter of oxygen in our body. They play a role also as defending our body. They can be also antibodies, defending antibodies. They can make some structures also. They can be enzymes like hydrogen peroxidase that will catalyze the hydrogen peroxide into water and oxygen, which is very nice because it will catalyze this toxin. Hydrogen peroxide is very toxic and you will catalyze it into water and oxygen. So yeah, they have several functions for the protein. Transport, structural, play a role also in the contraction of the muscle, myosin, actin, transporter. So the basic, the amino acid I told you, it's, what is it? It's an amino group plus an acidic group. And what is the acidic group? The functional group. If you look at your previous table, I go back to my table. Let's go back. Here you go. I am. Which one is acidic? It's this guy. This is the acidic group. So yes, Imani, thank you. It's the carboxyl group. All right, so it's an amino group plus a carboxyl group. This is what is an amino acid mean. It's an amino group, and let's look at the structure. We are going to see that it is an amino group that is with a carboxyl group. Look at it. That's the amino group. We are right, right? And this is my carboxyl group. And the carbon that is bound to the amino group and the carboxyl group and sample hydrogen, then an R, it's what we call the alpha carbon. Alpha carbon is the carbon that is bound to 4 different structures. One of these 2 structures, one of them is an amino group, the other one is carboxyl group. This is what is an alpha carbon in amino acid. All the time you are going to look at where is your alpha carbon when you have to deal with an amino acid. And your alpha carbon is the one that is attached to an amino group and carboxyl group and hydrogen, and what is left is your R. So all the amino acid, this is the major structure. This is the general structure for an amino acid. They all share that, but they differ by what? The R. The R can be a single molecule of hydrogen like the glycine over here. It can be a methane like alanine. It can be, look at this molecule, CH3, double methane with the CH, that's a valine. I'm not going to ask you to memorize all these 20 amino acids, but I want you to know the major function. The first thing is to discover where is your alpha carbon. That's my alpha carbon because this is my carboxyl group, this is my amino group, and that's my H. And what is left is your R. And this is exactly what we done in the lab to discover, if you remember, Amani and Kate and Yvonne, we work on that and came to determine the R for each amino acid. And whatever it's here in blue, this is our R group. This is how they differ from each other. And this R can put them in different classification depending of this R. If this R is polar, negatively charged, positively charged, look at this. is a positively charged. Sorry, this is what I am talking about. Look at this negative here. This is negatively charged. This is positively charged. It can be charged an amino acid. It can be negative or positive. Depend on the R. If it's positively charged or negatively charged. It can be also non-polar. Look at that. That's non-polar. There are no polarities over here. They can be polar, but no charge. This is polar. This is also non-polar. Depend on the R. How we form a peptide? The smallest peptide that we can form is a dipeptide. And after that, we don't call it tripeptide, tetrapeptide, pentapeptide. We call it polypeptide. That's it. We call it, it's a protein. But when it's only two amino acids together, it's a D for two, dipeptide. Dipeptide. How it's formed? And they are forming this peptidic bonding. Remember, when glycerol bond to lipids, the H of the glycerol give me the H, and the carboxyl group of the fatty acid will give me the OH to make a molecule of water, and therefore I make what we call ester, E-S-T-E-R, bonding. Ester bonding for carbohydrate to polysaccharide as well as disaccharide. It's also by condensation or dehydration. I make a molecule of water. How? When carbon of monosaccharide will give me the hydroxyl group, and the other carbon of the other monosaccharide will give me the hydrogen to make a molecule of water. Here, it's the same thing. It's also dehydration or condensation reactions. When amino acid will give me the hydroxyl group, which one is the carboxyl group of it? Because this is the one that has H. And the other amino acid will give me the hydrogen from the amino group. And therefore, I will make my molecule of water. And this is peptide bonding. So I bond this amino acid with this amino acid, forming molecule of water by peptide bonding. And this is how I am going to make this big polymer, like a glycine with isoleucine, one molecule of water formed. That means the carboxyl group of the glycine is giving me the OH, and the amino group of isoleucine will giving me the H, etc., 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 etc. And this is the primary, when it's like that, that's the primary structure of protein. And how this protein is expressed, of course, it's under the dedicated by the DNA. The DNA is the one that will give that this glycine will be in contact with this isoleucine. And this isoleucine is contact with the valine. And this valine is in contact with glutamine. And glutamine with acidic, with cysteine, etc., with alanine. This position of this typical amino acids is dedicated by the DNA. It's the one that will tell the amino acid, okay, be there. If I change one amino acid, I change completely the function and the characteristic for this protein. For example, we have, you all heard about the sickle disease, all right? the blood is not circulating normally. And this is how their hemoglobin look like. This is the normal hemoglobin, and the red cells, they look like the donut normal, right? Smooth, round, like a donut hot, so without a hole, anyway. And they carry the oxygen normal, no problem. And sickle disease, they cannot move easily through the blood vessels. It's very hard. And therefore, by blocking the flow of the blood to the lump and organ, they didn't get enough oxygen. And this is very deadly disease. And when they look at the genes that, the wild type of the hemoglobin, they look at this amino acid in the position 6. It's completely different. And when we talk about the gene expression, we will come back and see what's happened. How come this not being expressed at this glue amino acid? How come? How come we have valine instead of glutamine as an amino acid in our hemoglobin? And this completely, one amino acid, can you believe, changed completely? It makes us have disease called sickle cell disease. One amino acid was not being expressed. Instead, we have expression of another amino acid, change completely, cause this disease. This is very, very important. And then when we look at the gene expression, actually, you are going to see only one nitrogenous base that has been changed, one. And therefore, the expression of the amino acid completely changed. Secondary structures, what is it? We have two types. We have alpha and beta pleated sheets. Alpha pleated sheets, it's when this amino acid, if you look at it, this is alpha, it's like an helix, right? And the way it is like this amino acid will bond to the next amino acid, not far away, by what we call hydrogen bonding. A lot of hydrogen bonding exists on the alpha helix. And because this hydrogen bonding between the carbonyl form of the amino group and the carboxyl, which is the carboxyl group of the amino acid group, and the peptide bond, black bond, this will create this like folding on itself, the protein. That's the secondary. The tertiary, what is it? Instead to have also hydrogen bonding, we have other bonding. We have hydrogen bonding, but we make also a sulfur bridge. And the protein also interact not only between the amino group and the carboxyl group of the other amino acid, there are interactions on the R group. The R group also will interact with another R group of the other amino acid, creating a 3D dimensions shape of the protein. So we have in the tertiary, we have the hydrogen bonding, but we have the R of this amino acid that interact with the other R. And we have a lot of disulfide linkage on it. We have also quaternary. And actually quaternary, what is it? If you look at it very, you see that there are 2 to 4 like polypeptides. This is a beta shin, beta 1, beta 2, alpha 1, alpha 2, and they are bonding with each other. So in the quaternary, therefore, you have the protein the structure is consisting of more than 1 polypeptide. The tertiary, you have the hydrogen bonding between the amino and of the one amino acid with the carbonyl of the other amino acid. That forget about the peptide bonding. They are going to bond by hydrogen bonding, right? But also the R is going to bond with another R of another amino acid. For alpha, for the secondary, either alpha or beta, this is only hydrogen bonding on it. So we'll move to the next macromolecule, which is nucleic acid, DNA and RNA. DNA, it's your material genetic. RNA can be the messenger of RNA that play a role in the gene expression or the protein synthesis. We have ribosomal RNA that make the ribosome. If you remember, the ribosome are the factory for protein. And we have also transfer RNA that play a role on transporting the correct amino acid to the correct place to make the protein. So the RNA, either ribosomal or messenger or transfer, it play a role to transfer the information, the genetic information, into a protein, to get expressed into protein. So the genetic information came from the DNA, and he get expressed into protein via the help of the RNA, either ribosomal, messenger, or transfer. Nucleic acid is also a polymer. What is the micromolecule that make this nucleic acid, this micromolecule? We call it nucleotide. Nucleotide, exactly, Amani. This is a nucleotide. Nucleotide. Look at the nucleotide together. Me and you right now is made of what? What is that, Amani? What is it? It have a P on it. What group is that? Phosphate group. Phosphate group, correct. And what is it? It's a CHO. Looking like that. What is it? No, it's made of a lot of carbons, a lot of hydrogen, and a lot of oxygen. And it's single ring. What is it? It's a sugar. 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 Sugar can be either 6 carbon, that's an hexose, or 5 carbon, that's a ribose. Sugar. Memorize those. So he's made of phosphate group, a sugar, ribose, since it's a carbon. Let's see if it's 5 carbon. 1 carbon, 2, 3, 4, 5. Correct. 5 carbon. And this, I'm not expecting you to know it, but this is the nitrogenous bases. Remember DNA and RNA? They are all made of a sugar phosphate group and nitrogenous base. The nitrogenous base for the RNA are nitrogenous base for the DNA. What are they? You know it since the high school, dears. It's the adenine, guanine, cytosine, and thymine. That are the nitrogenous base. Cytosine and thymine are pyrimidine, one single ring. Pairing is double ring. There are 2 double ring, adenine and guanine. And we have 3 pyrimidine, cytosine, thymine, and uracil. RNA does not have thymine. It has uracil. Yes, adenine pair base with thymine in the DNA, but the adenine pair with uracil in the RNA. Cytosine pair with guanine. Do you realize like it's always pyrimidine base with the pairing base? You don't have pairing or pyrimidine. If you have pyrimidine, it's going to be too narrow, and if you have pairing with pairing, it's going to be too wild. So it's always pyrimidine with the pairing, and we will study that when we study the DNA. So the nucleic acid is made of a base, nitrogenous base. It could be, this base is 2 ring. Is it pairing or pyrimidine? That's a purine over here. It could be also pyrimidine, sugar and phosphate group. Sugar, there are 2 types of sugar, deoxyribose or ribose. If you look at your DNA, did you see your hydroxyl group over here? It doesn't exist. He got deoxidized. This is why we call DNA deoxyribonucleic acid, because the sugar that is there is deoxyribose. It is in the carbon 2. Notice that you have hydroxyl group on carbon 2. It doesn't have hydroxyl group on carbon 2. This is a big difference between the RNA and DNA. So recapitulations. I want everybody to write it down. DNA, what we have on it. Write it down, everybody. We have adenine, thymine, cytosine, guanine, and the sugar is deoxyribose. RNA, adenine, uracil, cytosine, guanine, and ribose. And both of them have a phosphate group. Okay? One thing that I want you to notice, and this is, we are going to name those carbons over here with a prime. If you look at it, you didn't notice that they put carbon and they put little prime on it over here? Why I done that? Why I put 3 prime, 2 prime, 5 prime? Why I put prime? Why you just put carbon 5, carbon 3, carbon 2? Why I have to put prime? Oh my God, I will give extra credit to whoever answered this question. Come on, guys. Why I put this prime on this carbon? Why this carbon over here in this corner? I put prime. Very easy. I give you a few seconds and they will answer. If I answer, no one will get the credit. Look at it very well. Okay, Amani, go. I'm listening. Amani, go. Why do we put on this carbon prime? Oh, your mic is loaded. Go ahead.
So when you read DNA and you have like a double, so when you have 5 carbon in the prime and then 3 carbon in the prime, that's reading how your nucleotides will read on a chain. So depending on where your chain starts.
No, the answer it's so easy, Amani. No. And Kate, no, also. Shall I answer? Oh my God, you're going to kill it. You're going to say, oh my God, no way. It's so easy. Come on. Which source of carbon you have there? Where do you see carbon? Other than I am close. This is a hand. Other than in this sugar. Where did you see it also? So if I said carbon 5, or I said 5 prime carbon, you are going to identify between which carbon to which carbon. Oh my God, easy than that? Hello? I'm amazed. Yvonne, go ahead.
Is
Yvonne.
it the RNA?
No.
And
Amani, go.
No,
No, it's not. It's not.
Is it just because it's the 5th carbon?
Is it what?
The 5th carbon?
No. Who was talking? Kim, go ahead. When you stop talking, stop your mic. Yvonne, no. Amani, no. Kate, no. Kim, do you got it? Oh my God. I swear I answered to the question when I was talking.
Hydrocarbon.
Oh. Yvonne, you have to raise your hand before you talk, and the answer is no. Is no. All right, I am going to give you the answers. All right, you are ready? Oh my God, you're going to kill me. It's so easy. Come on. I don't know why you think that the question was so difficult. Well, actually, you know the answers. We have other source of carbons molecules here in this nucleotide. It's not only this sugar, but also we have the nitrogenous base here. We have some carbons here, right? It's a ring of carbon. We have carbon here, we have carbon here. Where to differentiate between those carbons and those carbons, we put prime on the sugar. So when I've said carbon 2 prime, I mean the carbon that is in the sugar. When I said carbon 2, that means the carbon that is in nitrogenous base. Did you got it? Because those nitrogenous base, they are also have some carbons on it. Am I right? Right, they have some carbons. Did you see them? So to differentiate between those carbons and those carbons that are in the sugar, we put prime. Then Amani, yes, this is why we should that the nucleotide go from 6 prime to 5 prime to 3 prime. Because my bonding, which is the phosphodiesterase that is going to bond this nucleotide to another nucleotide, it's always between the phosphate group of one nucleotide with the 5 prime carbon of the next nucleotide. And if I look at again, let's go back. Where my phosphate group exists is attached to where? Which carbon? Read the slide. Where my phosphate group is attached. Amani? Come on. Just read the slide to define now, and you put prime, Amani, because here too, when you said my phosphate group is attached to the 5 carbon 5, which carbon? 5 prime. It's just prime that we put here to differentiate those carbons from those carbons that exist in this either single ring or double ring of my nitrogenous base. Did you get it? I told you the answers was so easy. So phosphate group, it's always to the 5' carbon. So this nucleotide will bind to another nucleotide, and the way it's going to do it is, if you look at it, come on, it's my 5' phosphate group, 5' will bind to the hydroxyl group of my 3' all the time of the sugar. It's always involving sugar. And its phosphate group with the next one. So this 3' will bind to the 5' via the phosphate group, and this 3' will bind to the 5' via the phosphate group, and this 3' will to the phosphate group via the 5' carbon. And in the end, and they think this is what Amino want to say, in the end what I have this 5' group that is completely free. He's not attached to any nucleotide. You see, it is free. And they have this 3' group is also free. So the directions of my nucleic acid, it's 5', 3'. And this is what Amani tried to actually, and this is, she answered to very complicated questions, and it was not my question. My question was why I put prime on those carbons numbers. It's just to differentiate them from other carbons that exist in the nitrogenous base. And why I call this nucleotide, this direction's 5', 3', because my 5' is not engaged to any bonding, and my 3' in the other side is not also engaged in any. So it is 5', 3'. Yes, Christopher? Yes, sir.
I'm still a little bit confused. I'm not sure, like, I don't know, I'm still confused on the question. So you're saying why do they have prime? Why are they called prime? I'm still not sure. Their answer, it's like a little bit confusing to me. Is there any way like you could like break it down?
No. Why we call it prime? Why we call this carbon 5'? Why any carbon in my sugar, I put this prime on it? It's just to identify those carbons from the other carbon, from those carbons. Because those also are carbon over here. So when I said prime, I mean immediately that is the carbon that is related to my sugar, not the carbon related to my nitrogenous base.
Oh, so it's basically just like making sure you understand that there are two different things, not are the same one. Okay, thank you.
Exactly, you got it, Chris. So when we put prime, that means those carbon are related to my sugar, not related to the nitrogenous base. This is very common to all what we use in science. When we use the word prime, immediately we know that this carbon is regarding the sugar, not regarding those nitrogenous base, because those also are carbons over here. All right? Okay, so, and this bonding that we made, we call that phosphodiester bonding. So phosphodiester bonding is the bonding that exists between the carbon 3' and the carbon 5' via the phosphate group. This is why we call it phospho and diester bonding. Phosphodiester. This is how I make my chain of nucleic acid, one chain. All right? How I make this pairing. So this is a pairing base is going to bound to another pyrimidine base. All right? By hydrogen bonding, and they will have this pair base, thymine bound to adenine and cytosine bound to guanine. And this is, so here it's much easier. So this is my first chain. If you look at it, this is my 3' is bonding to my 5' via the phosphate group, correct? This yellow thing. And then again, this is my 5', my 3' bonding to the 5'. And again, this is 3' to the 5'. Okay? So this, each time this yellow circles that you have here, this is my phosphodiesterase bonding. This is how this nucleotide is bound to the nucleotide. And then also this nucleotide, which is here thymine, because this is what I call nucleotide, is a nitrogenous base plus sugar plus phosphate group. So this entire nucleotide will bond to the other nucleotide over here via hydrogen bonding by pair base. And because of this pairing, I make a double-strand DNA. And it's also going in an anti-parallel way. So this 5' will bond with the 3'. If this is go 5', 3', this strand will go 3', 5'. It's an anti-parallel. Okay, so we are going to do some practice over here. I want you to tell me, what is this molecule? The first thing you discover is what were the functional groups on it. What is this molecule? Sugar, amino acid, sugar. Yes, Amani, this is an amino acid. This is an amino group and an acid. Yes, Christopher, correct. Now, we are moving to this molecule. You have glycerol, you have a phosphate group, and you have 2 fatty acids over here. So which molecule is that? Which one have a glycerol, have a phosphate group, and fatty acid on it? This is one fatty acid, and this is one fatty acid over here. Which molecule is that? Which molecule have a glycerol, phosphate group, and fatty acid? Phospholipid, yes, Amani. Thank you. Then we will move to the next one. What is it? Chem. This is a molecule that you have some hydroxyl group, and this hydroxyl group over here, this is also we have carbonyl group and a lot of hydroxyl group. 1, 2, 3, 6 carbons, all right? And so it's a base of C6 H12O6. Which one is have this formula, C6H12O6? Sugar, exactly, Yvonne. This is a sugar. You have a carbonyl group and hydroxyl group. And which type of carbonyl is that? It's sequestered between 2 carbon. And since it is sugar, we call it ketose. Ketose, it's a ketone. It's sequestered between 2 carbon. Okay? This is an open structure of glucose. And this one is also carbonyl and a lot of hydroxyl group. Carbonyl and hydroxyl group. That's the carbonyl group over here. All right, so this is also a sugar. Sugar. Okay. Now, we move to the next one. We have sugar, and we have a phosphate group, and we have a nitrogenous base. Nitrogenous base. Which molecule is that? We just saw it. Nucleotide. Thank you, Amani. It is a nucleotide. You have a pyrimidine Is it perin or pyrimidine? This one, I have only one ring, so it is a pyrimidine. Okay. Now, this molecule which have four ring, fuse ring. Which one have a four fuse ring? When you have four rings fuse with each other, which molecules? What we call it? Amani, you raise your hand. What is it? Yeah, it could be. That's a steroids. It's a cortisol. Okay, that's a steroid. All what I want you to answer is a steroid. This is a lipid. It's a four fuse ring. One false fuse ring. 1, 2, 3, 4. All right. Now be careful on this one. What is it? You have a lot of nitrogens. And this is why it starts by nitro. It have a lot of nitrogens. So we call it nitrogenous base. And this one, is it a pyrin or pyrimidine? Nitrogenous base. Exactly. Thank you. And this is a purine nitrogenous base because this is double ring. You have a lot of nitrogens on it and it's double ring. This one is also a nitrogenous base, but it is a pyrimidine. It's only one ring. All right. Okay, we move to the next one. Kate, I need to hear from you. What is that? We have a carbonyl group, of course. You see this ring here, if you remember, this is our carbonyl group, and a lot of hydroxyl. Carbonyl, a lot of hydroxyl. Which molecule have carbonyl group and hydroxyl? The sugar. Exactly, Yvonne. Is that a maltose? I don't think so. It's not a maltose because I have a ribose. A maltose is it's a exose with another exose. I have a carbon 6 with a carbon 5, so it must not be a maltose. But it is a disaccharide, and they think it's a sucrose. Exactly, Kate. I think it's a sucrose because fructose is a 5 carbon. The other one, it's also a lot of nitrogen. So we call it a lot of nitrogen. It's called, we start by nitro. Phosphate, we put phosphate as, because it's a functional group, all right? So this is a nitro. It's also nitrogenous base, all right? And this is a pyrimidine nitrogenous base, exactly. Now I have, you see 5 prime, 3 prime? Immediately when you have 5 prime, this is I translate that, translated. You want to translate 5 prime, 3 prime to what? To protein? Amani, you want to translate that? Oh, to protein? We didn't done the gene expression yet. Yeah, I will come back to that and ask you when I done the gene expression. What Amani she's saying, because it's amino acid, is 3 of them will be called in to an amino acid. And the way we called it, we are going to start by August, which is AUG, which is the start codon, which is the methionine, the codon. Yeah, no, we are not there. No, Amani, you are completely advanced there. Yeah, I can translate that. That's Amani mean. She wants to translate that into amino acid. This sequence can be translated on an amino acid. I can ask you one thing. Is it DNA sequence or RNA sequence? You can tell if it is DNA or RNA. It's RNA. Why, Amani? Can you explain it to your friend why you said all presence of U, which is uracil? Bravo. Now we are moving again to this one. Okay, what is it? I told you, when it's carbonyl plus a lot of hydroxyl, it is automatically, it's 1, 2, 3, it's a 6. Is it 6 or 5? This is 6. 6 carbon. I don't want you to tell me the name. That's an exose. Also, what you can tell, Kate, that's an hexose because it's thicker. It's a sugar. It's an hexose. Okay. What else? Is it aldehyde or ketose? Is it aldose or ketose? No. 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 Uh-oh. No. It's not a ketose. That's a ketose. The CO double bonding is sequestered between two carbon. Where is your CO? Can you circulate it, Amani? Why you get confused? Circulate my C double bonding O, which is my carbonyl group. Circulate it. Amani, can you circulate it? Yvonne, can you, anybody, can you circulate for me CO? Why? Just put a circle around it. Exactly. That's my CO. Is it in the end or sequestered between carbon and carbon? It's in the end. So it is an aldehyde form. Exactly. And it's an aldehyde form. And since it is sugar, we called it aldose. Aldose, double bond in CO. C, double bond in O. That's my carbonyl group. And this is an aldehyde form. So therefore, it's an aldose. This is a ketose. This, it's my ketose. It's sequestered between two carbons, one over here and one over here. That's a ketose. This one is in the end. So this is an aldose. Okay? Let's move on. What is that? Methionine, serine, alanine, arginine, phenylalanine, glutamine, alanine, etc., etc. What is that? This is a chain of amino acid, and they are all bound by a peptide bonding, an amino acid chain. What we call it? Peptide. Okay, or polypeptide, or just a protein. Okay. Now, all right. I want everybody to focus. What is that? Each one of them is. This is a sugar, yes, we know that this is a sugar. This is a molecule of sugar. Each one of them is what? It's a monomer of what? What we call it? Monosaccharide. Monosaccharide. This is one mono. What did you see hydrolysis, dear? This is a monosaccharide. Actually, I made a polysaccharide, and this is what we call glycosidic linkage. And this is made by, not hydrolyzed case, it's made by dehydration or condensation. All right? Dehydration synthesis, right. So if I want to ask you this polysaccharide, how many waters has been made here? How many molecules of water has been made? How many? 4, 3? Count how many glycosidic linkage. Is it 3? Is that right? 4. 1, 2, 3, 4. Correct. 4, Hailey. Amani, yes.
So I see that there's 4.
Count this last one.
Oh, okay. I'm confused.
Because this is glycosidic linkage over here. You don't see just, you see that is made in a glycosidic linkage.
Oh, okay.
So it is 4. Okay. Do you have any questions? Because we are done. I'm going to stop the recorder right now. And I'm going to